Okay, so this one is about chromosomes, and we already talked about chromosomes a little bit last week, and cell division. And I think the first slide is about something called telomeres. Okay, so cells, and it can be our cell that makes up the whole entire organism, or it can be cells in multicellular organisms. Naturally, cells are mortal. So mortal means like pretty much like everything in this world, right? So there is a time to be born, grow up, develop, and then die, right? So while we actually, count our age by years, cells also have kind of like internal clock. And this internal clock pretty much related to cell divisions, right? So after several divisions, cells actually stop, right? And again, that's time for them to die. Um, I think it was when it was in the 80s. That was a huge breakthrough when American scientists actually discovered that at the end of the chromosomes, there is a structure called telomere. Telomere is a section of DNA that doesn't code for anything, but every time when the cell divides, the telomere shortens, right? So shortens, 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 and when actually it's gone, right, then the cell dies. So telomere is considered like a protective cups. And basically looking at telomeres, you can estimate the age of the cell. Uh, when I said when actually that was discovered, the whole idea about telomeres was, okay, is it possible to harness this process somehow and actually do something about aging, because it's related to aging, right? So the older the cell, the shorter the telomeres, right? And it is related to aging for sure. And I don't know if you ever heard about this disease. This is called progeria. Guys, have you heard about this?
No.
You never seen this condition before? This is like when kids actually go into premature aging. So by age like, you know, 10, 12, they look quite old.
I thought that was just a movie.
No,
Like
it's
Benjamin
actually,
Button.
no, progeria, it's a real thing. It's a very, very rare genetic disease, but it's just a very real thing. So,
Oh yeah, I seen that.
yeah, and uh, the thing is that actually what accelerates aging inside their cells, and I think there's still no straightforward answer, but yes, uh, telomeres play a role in that. Okay, now can cells actually regulate the clock somehow, like control it? Uh, for example, cells that actually produce gametes, the sex cells, right? And these cells undergo a lot of divisions, especially in males, right? Sperm production, they actually can rebuild their telomeres, right? So they age as well, but much, much slower, right? And another thing that we're going to talk about later today is that cancer cells. And cancer cells actually become immortal. That's one of the issues with them. That what regular cells in the body are mortal, right? They die at a certain age. Cancer cells stay young, and somehow they are actually able to rebuild their telomeres and continue with the divisions. Okay. Now, cell division is first of all about proper division of genetic material, right? So it's important to split the cell in two, right? But before cell actually physically divides, it has to duplicate its DNA and prepare for division. So remember, DNA is packed into structures called chromosome. Chromosome is basically DNA plus proteins. And we talked about prokaryotic cells. Prokaryotic cells that don't have a nucleus, they have a single chromosome that is actually circular, right? And it's located in the area inside the cell called nucleoid. Eukaryotic cells have linear chromosomes, right? Not circular, but linear. And depends on the organism, the number varies, right? And chromosomes located inside their nucleus, right? So for prokaryotes, division is very, very simple. Please pay attention, that's gonna be on your midterm. Prokaryotic cells divide through what's called binary fission. So binary fission, the circular DNA just copies itself, right? Two circles. Once division of DNA is, sorry, once duplication is complete, cell just stretches and splits into, that's why I called binary by two fission, like split, okay? This type of reproduction is called asexual. Asexual means one organism can do the whole thing in terms of reproduction, okay? And two new daughter cells will be absolutely identical to original cell. Okay, in eukaryotic cells, it's different, okay? Now, one more time, depends on the cell, okay? When the cell is formed, right? It enters the phase called gap one. This is the whole like cell cycle, so I'm gonna go over it. So that's when cell carries out its normal function. If you talk about humans, if you talk about a bunch of other mammals, I believe some cells never go into division, right? So what you can see, gap one is when cell grows and divides, right? And this cell, sorry, when the cell grows and functions, and the cell can go into division. But you see G0, G0 means cells never divide. And if you talk about us, in our body, two types of cells actually don't divide. Muscle cells and neurons. Okay, in the nervous system, you have two types of cells. You have neurons that don't divide and cells that are called neuroglia. Neuroglia are capable of division through your whole life, but neurons actually don't. So you're born with like 85 billions of them, and what can happen to them? You can only lose them. What do you think? What actually, what usually kills your neurons? And if they're gone, they're gone. Talk to me, guys.
Wait, what did you say?
Isn't it usually chemicals?
Like, no, question was, like I was saying that we don't get new neurons, we only lose them, right? And you lose them pretty much every day, right? So my question was, what do you think, what kills your neurons?
Your urines, like when you pee.
No, neurons. Your brain cells, Ingrid.
Oh, what
Oh, okay.
neurons?
What?
What kills neurons?
What kills your brain cells?
Brain damage?
Drugs.
Drugs. Yes.
Yeah.
What else? What else, Fedora?
I was gonna say drugs or alcohol.
Drugs.
Yeah.
Yeah. But I'm saying something that happens every day.
happens every day.
Yeah, I hope no drugs and alcohol every day.
Like what you eat? Nutrients?
Your phone that's at the
Your
screen.
phone, I don't think your phone
I
really
hope
kills
not.
your brain cells.
If that's the case with food.
Yes, Ivana, what do you think?
Stress.
Stress, exactly.
Oh, really?
Absolutely, yes. Stress, that's what kills your neurons. I mean, a certain amount of stress in life, it's okay. It gives some, like, I believe it makes it interesting at times, right? But actually, for the health of your nervous system, you should really take everything easy. Tell you the truth. Like shake it off. Because unfortunately, neurons don't like, I mean, if they gone, they gone, right? So, what's really protects your neurons is learning, actually, right? That's what protects your brain. It helps to keep your brain young on the long run, right? Whenever you learn something new, whenever you learn new skills, like new things, learning new language actually is really, really great. Okay, what language would you like to learn?
Russian.
Russian, really? Why Russian?
Because everyone in my neighborhood speaks it, and it just, it would be very useful to know what they're saying.
Uh, I should say Russian is kind of like very unlogical language, to my opinion. Can
Yeah,
drive
it's
people
very.
crazy
I,
learning.
I think it's hard. It sounds hard.
I'm saying like it's hard in terms of logic. Like for example, let me give you example like Russian. If I ask somebody like, "Would you like to eat?" Um, and uh, like people usually say yes, people will say no. In Russian, people quite often answer yes, no. Literally, you translate it
No
yes, no,
way.
which actually means no. Yeah. So like, are you hungry? Do you want to eat? Yes, no.
Huh.
Yeah, so I mean, if you didn't like if you not really really like spend every day with Russians and like completely immersed in language, it can literally drive you crazy.
Yeah, seriously.
Mm-hmm. Okay, so where we are with cells being mortal and with cells that actually divide and cells that don't divide. As I said, neurons don't divide, muscle cells don't really divide, but other type of cells actually have a different rate of division. Okay, so cell that is actually the dividing cell gets ready. And the second phase is called S, which is DNA synthesis. So before cell goes into actual physical division, it has to duplicate a genetic material, right? So S stands for synthesis, and this is replication of the chromosomes. Then there gap 2 when cell grows a little bit more, and then cell goes into division, which is called mitosis. Okay, mitosis is the name for cell division. And mitosis, one more time, referred to separation of genetic material. Physical division of the cytoplasm is called cytokinesis. So that's what usually, that's what happens after actual mitosis. Let me write this for you on the whiteboard. Okay, so we talked about cell cycle, right? So it's G1, right? Then synthesis, then G2, then M, then cytokinesis. So M one more time stands for mitosis. Mitosis, by definition, is simple cell division. simple cell division? Simple cell division because the number of chromosomes in the daughter cells is the same as in original cell. So talking about humans, we have 46 chromosomes. So we start with 46, and after division, it will be the same number, 46 and 46. Okay, that's about mitosis. I think I didn't write it nicely. Mitosis. Mitosis. Okay, guys, clear? And that's what we're going to talk about today. We're going to talk about mitosis. Okay. Now, when the cell goes into mitosis, there's something called cell cycle control system. Cell cycle control system actually system that makes sure that no mistakes happen during this division. Any mistakes, specifically in duplication of genetic material or in proper separation, will affect cell function greatly. The cell should have exactly the same DNA, exactly the same amount of DNA as original cell. Okay, so first checkpoint is basically to check if DNA is damaged. There's sufficient nutrients for the cell. The second checkpoint, if DNA replicated properly, right? So synthesis, the whole synthesis phase went through well. And then assembly. Assembly is something that we're gonna see later on. Spindle assembly is system that evenly divides chromosomes between two new cells. Okay, so all the checkpoints control genetically, and any mutation that affects gene that controls cell division can actually result in cancer. Okay, so this is about chromosomal replication. So remember, chromosomes are made of DNA and DNA made of nucleotides, right? Nucleotides are built by complementary rule, right? T goes with A, G goes with C. So DNA synthesis is when the parent DNA molecule like unwinds, splits, right? Not entirely, like a region of the DNA opens up, and there are enzymes that actually make DNA copy, like they copy each strand, which will result in two identical DNA molecules, okay? So, and it's a surprisingly fast process, like thinking about so much DNA in the cell, but amazingly how fast DNA in the cell could be duplicated. That's what I was talking about. So remember, DNA is a double helix, right? Its region kind of like forms like a bubble, and on each strand will be separated, and its own copy will be built. Yes, so unwinding, separation, reconstruction, elongation. So this is the enzymes that, usually a couple of enzymes that do their copying of DNA, right? And because two strands, they kind of go in opposite directions, so the replication also goes in opposite direction. And it will eventually result in two identical copies. But still, nothing is completely, completely free of errors. So you see, DNA proofreading and error correction. So there's a system that basically checks the DNA and makes sure there are no mistakes in the copying. And again, no mistakes in separating. Like, any questions so far, guys? You with me?
I don't really understand any of this. So the mitosis is supposed to be the splitting of the cell.
Okay,
And
look, first of all, you joined late. Okay, so you
Okay.
joined late when I was
No,
giving
but Professor,
introduction.
you are going a little too fast.
Okay, sure. I'll slow down. So mitosis is division
Amore.
of cell. And for example, this slide says, right? Cells are not immortal. That's what we started with, right? Cells have a life cycle. There is time for the cell to be formed. There's time for the cell to grow. And there's time for cell to divide. I was also talking that not all cells, for example, in the human body, are capable of division, right? But if cells can divide, they can undergo mitosis, right? So the purpose of mitosis is growth and development, right? So that's how our body grew at least, from the very beginning during embryonic fetal development and early ages, right? So our body grows through mitosis, right? And in adult, there are tissues that actually undergo mitosis all the time, like your skin. For example, your top layer of skin, the one that you see from the outside, is actually made of dead cells. Why so? These dead cells protect your skin from the outside. But underneath, there are layers of cells that undergo mitosis, right? They undergo mitosis, they move from the bottom towards the top. And when they reach the top, they die, okay? And we constantly shed them. So your house dust, what you can see here, house dust is actually made of your dead skin cells, okay? Our hairs grow, they grow through the same process of mitosis, right? So for example, this guy shaves, right? He damages a bunch of cells and all the cells on the skin here will be replaced in no time, right? Sunburn kills a bunch of cells on the top layer of your skin, okay? So what happens? It peels off and regrows, right? So that's why mitosis is so important, okay? Especially in the tissues that provide protection from the outside. Does it make more sense now?
Yes.
Okay.
Yes.
So you see, mitosis replaces worn out old cells, but not only. For example, you see red blood cells replaced every 2-3 months. Cells that line intestines replaced every 3 weeks. Cells in your skin replaced every 4 weeks, I should say. Cells in your bones replaced more slowly, much, much slower. And as I said, your neurons die, right? So when cells die, there's usually a mechanism that triggers the cell death. You can say called suicide, and it's called apoptosis. So apoptosis is different cell. Okay, and by the way, one of their medications that actually developed to treat viral infection is to help the cells to commit suicide. The thing is that cells don't like viruses. They hate viruses for many good reasons. And I think I explained to you last week about viruses. So quite often, if cell is infected with a virus, to prevent further spread of it, the cell will prefer to commit suicide, to trigger apoptosis, rather die than to become source of infection. Okay, and as I said, new medications that treat viruses actually help cells to commit suicide once they are infected. Any questions here?
No.
Okay, awesome. So we're going to talk about mitosis, and then we're going to talk about another type of cell division. And then we're going to talk about a little bit more about mutations as well. Okay, so overview of mitosis. Overview of mitosis, this is their cell that is able to divide, right? And when cell is not dividing, non-dividing cell, even it's preparing for mitosis, but it's not in process of division, it's called interphase. Okay, interphase, non-dividing cell. Okay, so like the phase before actual mitosis. Now, what happens in mitosis? As I said, the most important thing is to duplicate genetic material properly and divide it properly in daughter cells. 1 second. So in the beginning of mitosis, chromosomes condense. They kind of like shrink. They shrink, they thicken, and they become more visible. And they look like letters X. This is beginning of mitosis. And letter X is pretty much due to the idea that one chromosome and its own copy attached, right? So remember, our chromosomes are linear, right? So they don't really look like letters X. Letter X indicates there are actually 2 chromosomes, right? So these 2 chromosomes connected to each other will be aligned in the center of the cell. And centrosomes, organelle that is responsible for division, will form something called spindle of division. Spindle are fibers, protein fibers, that help to physically separate chromosomes during mitosis. So once they align in the center, one of the fibers from each side will attach to one of their twins here and then pull them apart. They will pulled apart, 2 new nuclei will be formed, and then cytokinesis will finish the physical division of the cell. This is overview. Now we're going to talk about step after step, so it will make more sense to you. Okay, so that's what I was talking about. Chromosomes are linear, right? So this is one chromosome. During synthesis, this chromosome will be replicated. So there are like 2 twins, right? The original one and its twin. These 2 twins are called sister chromatids. That's what actually gives this, again, appearance of X's, right? Chromosome and its sister. They condense, again, they kind of like shrink and thicken, and that's how they look at the time of mitosis. This is the only time when they look like that, okay? So chromosomes literally visible only during mitosis. And no genetic tests when, you have to take a look. And the chromosomes, you actually have to bring cell into division because only during division, you can actually tell them apart and count them and study them, okay? And another thing that I mentioned, spindle. So spindle are protein fibers that able to attach to one of the chromatid sisters, right? And pull them apart during mitosis. Okay, so we're gonna talk about the phases here. And the first phase is, between mitosis is called interphase, okay? So interphase, when cell is growing, and one cell is preparing for mitosis, and when the cell actually replicates its genetic material. First phase of mitosis is called prophase. Prophase, nuclear membrane breaks down, chromosomes visible. Again, you see two sister chromatids attached. What attaches them is called centromere. Centromere is a structure that keeps them together, holds these two sister chromatids together. These are centromeres, which are made of centrioles, right? Centrioles move to the opposite poles of the cell and produce spindle fibers. So this is prophase. Metaphase, chromosomes align in their equator, like center of the cell, right? And each sister chromatid actually faces either one of the poles, right? The fibers attach to their chromatids, right? And in next phase called anaphase, these fibers pull them apart, right? Separate. This is anaphase. Telophase, chromosomes are separated, two nuclear envelopes are formed, right? And to complete division, cytokinesis will physically divide cells into two. I want to show you something here to make it a little bit more clear.
It undermines the writer's message, and the word choice is bland.
Mitosis. In this module, you will learn about mitosis. Mitosis is a process of cell division that makes possible regeneration of body parts. To elaborate, mitosis is the separation of nuclear chromosomes into two identical daughter nuclei. It is divided into four key phases: prophase, metaphase, anaphase, and telophase. An additional phase called cytokinesis occurs after the complete nuclear division. Before a dividing cell enters mitosis, it undergoes a period of growth and rest. This period is called interphase. In interphase, the genetic material of a cell increases. Prophase. During prophase, the chromatin condenses and becomes short and thick to form chromosomes. Each chromosome has two sister chromatids joined together at the centromere. The nucleolus disappears and the nuclear membrane starts to break down. The centrosomes, each with a pair of centrioles, move apart to the opposite poles, forming a spindle between them. Metaphase. During metaphase, microtubules of the spindle interact with chromosomes. This causes the chromosomes to move and align along the middle of the cell or the equatorial plate. Sister chromatids are held at the centromere, confirming the presence of protein structures called kinetochore at the surface. The kinetochore holds the chromosomes and attaches them to the spindle. Anaphase. During anaphase, the sister chromatids separate at the kinetochores, and the daughter chromosomes, along with the divided centromere, move towards the opposite poles as the microtubule shortens. The poles of the spindle also move apart and help to separate the chromosomes. As the chromosomes separate completely into two sets, one at each pole, anaphase is considered complete. Telophase. During telophase, the daughter chromosomes arrive at the pole and the chromatin starts to decoil. Also, the nuclei reform along with nucleoli. This triggers the reformation of the nuclear membrane. Cytokinesis. Cytokinesis is the partition of the cytoplasm. In animals, a cleavage furrow appears in the plasma membrane, which deepens and divides the cytoplasm into two daughter cells, each containing a nucleus. In plants, new cell wall formation is triggered at the center of the cell by the formation of a cell plate. The cell plate grows outwards till it meets the existing wall, resulting in two daughter cells. In this module, you have learned mitosis is the process of
Okay, guys, do you want me to go one more time over phases of mitosis?
Yeah.
Yeah, just one more time, you know.
You know, I can do it.
Thank
So,
you.
okay, one more time. Once DNA is duplicated, right? So each chromosome duplicates itself, right? And at this point, I know it's confusing, but at this point, when you have chromosome and its own copy, they call sister chromatids. Not really chromosomes, but sister chromatids. When they separate, they're chromosomes again. When they kind of like stuck together, they're sister chromatids. They're connected by something called centromere. So the whole idea of mitosis is to separate them evenly into two new cells, right? So interphase, when cell grows and when cell duplicates its genetic material before mitosis, that's interphase. When cell enters into division, into mitosis, first step, just to break down the nucleus, right? Because, you know, you have to separate this chromosome somehow, right? Chromosomes become like, they kind of like free in the cytoplasm, right? And centromere, which is their organelle in charge of division, breaks down into two pieces called centrioles. So one centriole goes to their, let's say, north, another goes to south, and they produce fibers. You saw in the video the name kinetochore. Don't really worry about this name. Just division, like spindle division fibers. That's called a spindle. Spindle means a system of protein fibers that actually will pull the sister chromatids apart, right? So end of each fiber will attach to one of the sister chromatids, right? This is metaphase in the middle of the cell. Anaphase, the fibers pull them apart. Once they reach the poles, two nuclear envelopes are formed. This is called telophase. And the last thing is to physically separate them into two new cells. That's cytokinesis. How about now? Makes more sense?
Yeah.
Okay, so something that is related to mitosis and something that people don't really understand much is that I told you cells are mortal, right? Normal cells, healthy cells are mortal. So after a number of divisions, they usually die and they replaced by new cells, right? I also told you that division, mitosis, is controlled by a number of genes, right? So these genes basically regulate when it's time to grow, when it's time to divide. They check duplication of DNA, they check how cells separate. If there is a mutation in genes that regulate cell division, the division becomes uncontrollable, okay? So cells decide on their own when to divide, how to divide. And what happens in cancer is cells eventually don't resemble the original cells in terms of shape, in terms of structure, in terms of function, right? They don't collaborate with other cells as well. They don't respond to any signals, right? So these cells have their own agenda in the body. And another thing, they become immortal. Unlike normal cells that I told you, they're immortal, cancer cells become immortal. And cancer is considered aging disease. Why aging? Because throughout the years, people can accumulate mutations that can actually cause cancer. If person in young age develops cancer, very likely that was like already genetically inherited from the very beginning. Okay. So my question to you, what can cause cancer? Like, what do you know about causes of cancer?
The causes of cancer could either be familial.
Yes, genetic, yes.
Could also be
Radiation?
found in like
No.
some foods.
Could be environmental, such as smoking, drinking.
The sun.
Absolutely, son. What else?
But
Like
most
lifestyle factors, like how you look, like what you eat.
Yeah.
Radiation?
Radiation, absolutely, radiation. But what else, guys?
ill health.
Okay, I'm sure you heard. I'm sure you heard about what is
HPV.
HPV?
Human papilloma virus.
Yes, human papilloma virus. You see,
Get a vaccine against that.
virus. Gardasil, that's a vaccine. Very good. But this virus is associated with what type of cancer?
It's associated with
Cervical.
Yep. Cervical cancer. It's actually also can be associated with their throat cancer as well. Okay. So what viruses do? So let's say this is the cell, this is the DNA, right? Remember, I told you what virus is capable of doing. They insert the genetic material inside the cell's own DNA. Remember that? You guys remember? I told you this last week when we talked about viruses, what viruses can do, right? So let's say this is the gene that controls cell division, right? So if virus basically inserts its own genetic DNA, so this is viral DNA, right? This viral DNA will break the gene. So breaks gene that controls mitosis. Not viruses capable of doing that. That's why there are a number of viruses that are associated with cancer, right? Like HPV, hepatitis virus, some adenomaviruses as well, right? Not every single one does the same thing. But you see, mutation can break the DNA, right? Radiation can cause mutation that will break this DNA. Some other things can also break this DNA, but viruses physically do that, right? You see, they just insert themselves exactly where genes that control mitosis are, right? Breaking them that way. So, as I said, cell cancerous cells, right? Oh my gosh, sorry. Cancer cells over time, they undergo many divisions, and they stop even looking like original cells. And I told you, they have their own agenda. And what's the agenda? Is to grow, divide, and spread, right? So initially they grow in one particular area, right? And then they start spreading. Anybody knows the name, how cancer spreads? What's the name of cancer spreading to other places?
I think it's called. God, I forgot.
Yes, that's called metastasis, so, right? And when it spreads to other locations, right, and grows, it literally starts stealing resources from the body, right? I told you the cells don't cooperate with other cells, other tissues anymore, right? They only care about their own benefits, about their own well-being. And what usually causes death is that these cells start affecting function of vital organs, right? On top of causing pain, they pretty much destroy other tissues and organs, and that's what causes death from cancer. So my question to you is, yes, so you see, spreading. My question to you, how this could be treated? What's usually treatment for cancer?
Chemotherapy?
Yes, chemotherapy, exactly. But usually before chemotherapy.
Wait, Professor, can you repeat the question?
Um, what is a treatment for cancer before chemo?
Wouldn't they try to remove the cells if they can?
Yes, usually surgery, right? So they try to remove, yes, the tumor. And then, yes,
Immunotherapy,
Therapy?
yes, guys, go ahead.
some of them try to use immunotherapy, which is taking some, I guess, their blood or some of their immune cells and CAR T cells to fight their own cancer cells or something like that.
Yes, absolutely. The thing is that cancer cells are very sneaky and they actually try to trick immune system by pretending that they're okay. So immune system ignores them, right, and allows them to grow. Natural immune system has to fight them, right? Identify and fight them. So, but chemotherapy is actually therapy that is designed to kill cells that actually grow very fast. Tumor cells, they have very high rate of metabolism. They grow and divide very, very fast. So chemotherapy tries to slow down this division. And unfortunately, along the road, it actually affects healthy cells that also divide in high rate. They affect cells that produce hair, they affect skin, they affect blood cells. And quite often people who have chemo, what happens to them? They lose hair, they have serious issues with skin, and sometimes they need help with their blood production as well. Let me just show you something. I hope I can find this. One of my favorite videos, if I'll be able to find it. I hope I will be able to find it. Can we watch this one?
Providing a safe place to receive care is Valley's pledge to you. Whether you're experiencing a medical emergency, undergoing a planned procedure or surgery, receiving a home care visit, or have a scheduled appointment, we're taking every precaution to ensure your safety.
This next story is about a remarkable treatment for leukemia that has astounded a family in Philadelphia. 7-year-old Emily Whitehead endured the disease for 2 years. She'd exhausted every conventional treatment. Her parents were told she was close to death. But there was one last hope, an experimental treatment that amazed everyone when it worked. Eric Sorensen reports.
Her mom first noticed it, Emily's bruising, then bleeding. She looked up the symptoms.
Bruising, gum bleeding, nosebleeds, and the first thing that pops up is leukemia.
leukemia, and for Emily, it was worse than that. Chemotherapy that helps most kids didn't work on her leukemia. She thinks her dog sensed the cancer was back.
She wouldn't like play with me or anything. She would just sit.
Emily was dying.
We knew it wasn't kind of the standard garden variety leukemia.
Desperate, Emily's parents rolled the dice on a new therapy to genetically modify her own immune cells.
You're going to get your T cells? What are they going to take away? Cancer. That's right.
T cells fight infection. Doctors removed Emily's T cells and re-engineered them using HIV with the disease-causing part removed and re-injected them to attack the leukemia.
They took us out in the hall and said, there is no room to get sicker. She is as sick as you can get. You know, they said, you should call your family in. There's a pretty good chance she won't be here in the morning.
At first, Emily got sicker, but then began to get better. You can see the T cells at work, the smaller cell attacking the bigger cancer cells.
This is a whole new way of treating cancer. And the idea is that we're redirecting the cells of the immune system against the cancer.
Cool.
8 months later, Emily is like her old self.
I thought we were witnessing a miracle.
Doctors seem just as amazed.
We can't find any leukemia in her body using the most sensitive tests that we have.
When he told us that news, I looked at Carrie and I said, she's going to get better.
For now, Emily is cancer-free. If she can stay that way for another 16 months, she'll be considered cured. Eric Sorensen, Global News, Washington.
Brush your teeth.
Okay. So guys, did you understand like what happened? Yeah?
Wait, but they use HIV? I don't
They
Okay.
injected HIV in her T cells.
Right.
Okay, so look, this is a T cell, right? This is very important cells of the immune system. The most important cells of the immune system. Remember when I was telling you about the viruses, that viruses are very specific to what cells they infect. This is one thing. Another thing is that viruses bring their genetic material into cell, right? So what they did, they took the HIV virus. Remember, I told you virus is just a piece of genetic material enclosed in a protein shell, right? So they took the viral RNA out and then they genetically modified it. So they left the piece of the virus that can enter cell. Remove the piece that can kill T cell, an attached piece that activates T cell against virus. So, remember last week we talked about biotechnology? So this is literally biotechnology in action, right? So they modified the HIV virus and the virus entered the cell, right? And no, one thing before it. So they took her blood, right? They collected her T cells. They infected these T cells with this modified virus. And then they inject these T cells back into her. And at the beginning, reaction was very, very bad. She developed fever and doctor said that like there's no way, no room anymore to get sicker. So she was so sick that the next step was just dying. And then exactly what happened, the miracle, right? It's really a miracle because these T cells kind of like woke up and began chasing, attacking, and destroying the virus, their leukemia cells. And that's what cured her. I know now that in these days, this therapy is available right now, but I think in these days they use other viruses. They don't use HIV virus, they use another virus that's also able to get into the T cells. And that's what actually treats cancer pretty much the same way.
Does she have HIV now? Or is it...
Andrea, they removed the one that can cause HIV. This is again a virus. It's like a virus. They use it as a key, right? because it's able to get inside the T cells, right? So to open the cell, but nothing more than that. So their pathological, the disease-causing part was removed. So that's the whole idea. Remember, I told you, genetical engineering is all depends on viruses, because viruses are the keys to bring in genes inside the cells. Okay? Okay, guys, so like a few more minutes and you take a break. So that was about treatment and reproduction. So the next part, let me see how many more we have meiosis.
Professor, all of these slideshows are on the BIO108.
Yes, every week I post them. Absolutely.
Thank you.
Okay, so what is reproduction generally? Reproduction is, by definition, is passing genetic material to offspring, right? Creating offspring with a combination of genetic material. So reproduction could be asexual. Asexual is when it's actually from one parent to offspring, right? And it's like cloning, literally like cloning, because the offspring will be genetically identical to their parents. This is more common, much more common, because like majority of organisms in this world actually reproduce asexually. Sexual reproduction is when there's a combination of genetic material from two parents, right? For this combination, organisms have to produce special cells. This special cells called gametes. Let me just show it to you. So you can say that like any cell, and usually we call this any cell that is not sex cell, we call this somatic, has two sets of genes, right? Like we have two sets of genes, half from mother, half from father. We talked about this. If set of gene is N is set of gene, so 2N means diploid. So all cells in our body are diploid, except for gametes. Gametes, which is the same as sex cells, which it could be ovum or sperm. They have only one set of genes, one N. They're called haploid. So you have one haploid plus another haploid, right? One N plus another one N. And here you go, 2N diploid, right? That's what happens during fertilization. Fertilization is literally joining genetic material from two parents, okay? And that's about sexual reproduction. So sexual reproduction is definitely preferable in terms of evolution, because it gives you what's called a combination. It gives you like more variations on genetic material, right? Now, some species, you see some plants can do both. They can do both sexual and asexual, okay? And sexual reproduction, in sexual reproduction, the organism has to produce these gametes or haploid cells. And this process of cell division that produces haploid cells is different from mitosis. Remember, mitosis, you have 46, you end up with 46. In this type of reproduction, you start with 46 and you end up with 23, right? And And this process is called meiosis or reduction division. Okay, guys, let's say 10 minutes break.
Yes, please.
Okay, guys, and we'll continue with the cell division. Okay, so we'll continue with meiosis. Okay, so meiosis is crucial for sexual reproduction, right? Because meiosis produces gametes, right? It takes a cell that was 46 chromosomes and turns into a cell that has 23, right? So in females, in males, meiosis is also called gametogenesis. So gametogenesis is production of gametes, right? And in males, it's called spermatogenesis. And in females, it's called it's double oogenesis. Okay. And one more time, the whole idea of meiosis is from 46 to 23, right? Now, meiosis and mitosis, they have a lot of things in common, right? But meiosis is actually a more complex process of cell division. And while mitosis happens throughout the whole entire life, meiosis begins, actual meiosis begins at puberty, right? Production of sex cell, ability to reproduce. So meiosis takes place in reproductive organs, which are called gonads. Basically, males testes and females are ovaries. And in animal world, there's still testes and ovaries. Okay. And as I said, it begins with 46 chromosomes. Just 1 second, guys. Ты можешь закончить, пожалуйста? It begins with 46 chromosomes, right? And remember, 46 is literally means 23 pairs, right? So each chromosome has a pair. There is a chromosome from mother, maternal, and there's a chromosome from father, right? So these two chromosomes, literally the same. The only difference is that they carry different genes, right? Maternal genes, they have maternal versions or alleles, and father chromosome, homologous chromosomes carries father's alleles, right? So remember, that's something from the last week I told you that homologous chromosomes can carry different versions of genes. So this is a gene for the shape of your ear. Mother can carry gene for round ear, and father can carry gene for pointy ear, something like that. And again, like in mitosis, they replicate, right? So they replicate, so you see their sister chromatids, right? And there are also additional sex chromosomes that we talked also, like X and Y. Now, the major difference, look, in meiosis, you start with 46, right? You duplicate it to 92. And then you have 4 cells by the end, and each cell will have 23. Remember, mitosis, 46 duplicate to 92, 46 and 46, right? Meiosis, 46, 92, 23, 23, 23, 23. Is it clear?
Just a question. Can the diploids, sorry, can the haploids become sister chromatids again?
No, haploids is the end result. No.
Okay, sorry about
Haploids are for fertilization, for reproduction. This is the end product, okay? Mitosis. Haploid results in haploid. Diploid and results in diploid. In meiosis, diploid results in haploid. Guys,
Okay.
is it clear?
Okay. Yeah.
Okay, the rest of you guys, please talk to me. Is it clear?
Yes.
Yeah. So again, why you need haploid cell? Because each parent can contribute only half of genetic material, nothing more than that. Okay, that's the idea, right? Because if you give diploid cell and another will give diploid cell, it's not going to make sense, right? Too much. So you have to give exactly half. Okay, so the most important thing in meiosis, and let me explain to you. The most important thing, and I'm just going slightly forward and then I go back. Okay, so this is mothers, this is fathers, right? So these two homologous chromosomes, and each chromosome basically is already duplicated, right? So duplicated, connected, they're sister chromatids. At the beginning of meiosis, in the first part of meiosis, okay, these two homologous chromosomes, mother, father, they are getting very, very close to each other, right? So they pretty much like fuse, right? They fuse, they form connections that call chiasmata, and they start exchanging pieces with each other. That's called crossing over, right? So what will happen, right? If you look here, so that's what you started with, and that's what you end with, right? So maternal and paternal chromosomes kind of like scramble and unscramble, right? And this actually creates combination of genes. So it's not that
Kind of like a randomizer.
it's absolutely random process. And this is the reason actually why children, when the same family, they look slightly different from each other, right? So look, this is like Kennedy family. You can tell that they're brothers, right? But they don't, not identical. This is the reason, actually crossing over is a reason why kids within the same family don't look alike. They look different. Yes.
Sometimes very different.
Sometimes very different. Absolutely. Yeah. Sometimes you can't even say that, you know, they are related, but sometimes, yes, it really depends. It really depends on the genes. But crossing over is pretty much what is meiosis about? To create combination of genes in each cell that it produces. Okay. So if I go back, interphase, right? And then prophase. So prophase 1, like in mitosis, you get duplicated number of chromosomes, and that's when their chiasmata and crossing over happens at the very, very beginning, right? And what you can see here in metaphase, when they align in the equator, you see that they're slightly different, right? So they already after crossing over, okay? So anaphase, they get separated. And then once meiosis 1 is completed, what you can see two diploid cells. Two diploid cells, each has 23 chromosomes, right? Connected to each other, like, so it has 23 pairs, right? Connected to each other, but they already scrambled, unscrambled through crossing over. So this is end of meiosis 1. In meiosis 2, all they have to do is just to separate. That's it. So you see each cell has diploid set, no more replication, nothing. They align in the middle, they separated, and by the end you have 4 cells that are haploid. Okay, so meiosis 1, duplicate, crossing over, produce 2 diploid cells. Meiosis 2, take these 2 diploid cells and separate genetic material further on to get 2 haploid. Any questions here?
Uh, I'll ask you later.
No, guys. I mean, no. It's okay. You can ask me anytime.
I don't even. I'll just read the book and ask you. I I hate having a learning disability. So firstly, the cells develop. I'm sorry. I'm just
Okay, so let me just put this way. You look at my mitosis, right? Mitosis of 46, right? So this 46 times 2 get 92, right? And then end result 46, 46. Are we good here?
yes.
Is it clear? That's mitosis. That's what we started with. Meiosis is you have to produce cells that will eventually have only 23 chromosomes. So you get 46, right? Duplicate. Sorry, then you take 46, you duplicate by 2, right? You get 92.
Yes.
And then you get 46 and 46, okay? This is meiosis 1. Then this 46 will divide to 23, 23, 23,
Okay.
Does it make more sense now?
Okay. 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 Yes.
Yeah. So mitosis, diploid to diploid. Meiosis from diploid to haploid.
Okay.
Guys, any more questions? Please talk to me. Everything is clear, nothing is clear.
Meiosis one is the top one, right? The top right one. That's 46 times 2 equals 92. That's diploid to haploid.
But first of all, you take diploid, you make 2 diploids, right? And that's from 2 diploids, you make 4 haploids.
Okay. All right. I'm starting to get it.
That's how sperm cells produced. Like, look, for example, if I go back here, guys, one more time. Any questions?
Somebody else ask a question, please.
Okay, so you see, next one is that males and females produce different way. Okay, so the thing is that in males, this is like classical meiosis, like from 46 to 23. Okay, so each diploid cell will produce 4 haploid cells. And in males, this process starts at puberty and continues pretty much through the whole entire life, right? In females, there is a window, right? Like a fertility window. What happens in females, first of all, is that egg cells, right? The oogenesis starts before birth. It starts before birth and it freezes approximately like it freezes before birth. So it starts like 5 months through fetal development, and before birth it stops, it freezes. And girls already born with their approximately 500,000 of egg cells, right? While males can produce, can't estimate how many through their lifetime. Females at birth already have only 500,000. Of course, they will never use this 500,000, but once they reach puberty, like age 12, something like that, half of them will be already gone, okay? And during life, they don't make more than approximately 50, only 50, no, why 50? More. Sorry, like 200 something mature egg cells, okay? And the process of meiosis stops when? When females stop producing egg cells? When they reach what?
Oh yes, I know this. I know this. It's God. Um. What's the word?
The word, I believe, is menopause.
Stephen.
Right?
Hello. Yes, you are right. Yes, that's menopause, right? So the window is actually between age 12, right?
Yeah.
To age 50. So something like 38 years for female, right? By the way, who knows what's the oldest woman known to give birth?
I don't know.
Anybody knows?
80 something.
80? No. I think younger.
50?
I think the oldest couple, I think she was 70, something like that. And he was 95. And I think they're from India. Yes. So I think that's the record actually. 95 for father and 70 something for mother. But again, usually females, like by late 50s, they already go into the menopause. Okay. And another thing is in females, remember in males, one diploid makes 4 haploid, right? In female, actually only 1 egg cell will be produced. So you see during meiosis 1 from diploid to diploid, the second cell will be discarded, right? That's called a polar body. That's the way to discard extra genetic material. And then from meiosis 2, right, it will be discarded again. Okay, so basically what happens, one diploid cell makes only 1 haploid cell. But the body puts a lot of resources into this cell. The cell is huge, right? And remember, the cell carries basically all the cellular contents for the embryo. So look, huge female egg cell and tiny sperm cell. Okay? Makes sense? You with me?
Yes.
Yes, all of you?
Yes. Yeah.
Okay.
Yes. Yeah.
So, what else I wanted to tell you is that, yeah, problem is that I told you females kind of limited in number of egg cells that they make, right? And usually, what do you think? What's the best age for having children for women?
I think maybe late 20s, 29, 30 maybe.
Mm. Younger.
Younger, 26.
The best age is actually from 20 to 30. It by like end of 30s, the number drops dramatically. Okay, so around 40, it doesn't mean that female can get pregnant.
Yeah.
And can have healthy children. Yes, they still can, but the chances are much, much lower actually to produce an egg cell and actually have a healthy child. Okay, and what are the benefits? So benefits is that, again, you have a combination of genetic material from 2 parents, right? And it's literally like throwing dice. So you don't know exactly what combination will be, like what exactly parents contribute, which comes again from crossing over that generates these variations in reassortment. So when they separate here during anaphase 1 and anaphase 2, you never know which way they go, right? which chromosomes will go together, which also increases variation, genetic variation in offspring, okay? So we can say that meiosis is definitely driving force for evolution. And as I said, it's really beneficial, but there's some limitations. So first of all, the biggest limitation is to find their mate, right? I'm not talking about humans. I'm talking about animal world, right? So if you are, let's say if you're a lizard, right? If you're a lizard and nature definitely gives you that drive for reproduction, right? You have to find a male lizard. Makes sense, right? But for some organisms, it's kind of like it's kind of hard, right? So for some organisms, they have an option actually of reproduction on their own. So these organisms are called hermaphrodites, right? Yes?
Yeah. Yes.
So like, for example, if you look at their, good example of hermaphrodites, you know who? Earthworms, right? So earthworm has an area where they can produce both sperm cells and egg cells, right? So like kind of like female part and kind of male part. I don't think they really have an issue finding a mate because there are a bunch of them out there, right? But just in case, can find another earthworm, here you go, right? And also for some organisms, they have to pay price. This is who? I'm sure you heard about Black Widow, right? What they do, females.
Um,
Come on. Okay, so this is who's male, who's female? Tell me. In this picture. The big one is who? Male or female?
Female.
Female. This is female. And this is male, right? And he approaches her really, really cautiously. Because of what? Because after he will fertilize her, what she's gonna do to him?
She's gonna eat him.
She's gonna eat him, exactly. Yeah. Why she's gonna eat him? Why?
Because I don't know. I don't.
Come on, guys. Hitori, why she's gonna eat him?
For strength? I don't know.
After he's gonna fertilize her, what she's gonna have to do? She has to lay eggs, right? Right?
Yes.
Now, to lay eggs, she needs what?
Oh, food? I don't know.
Exactly. She needs food. Absolutely. She's gonna be very, very hungry, right? So if he can run away from her fast enough, she's gonna grab him and have him for lunch to get some proteins to lay eggs. Have you heard about praying mantis? What females do?
Yes.
What they do? Yes, Catherine. Right.
After mating, the woman bites the head off and eats the body.
Yes, she's gonna bite the head off. Pretty much for the same reason, right? Okay, to get some energy. So you see, it's kind of like a tricky business, actually, risky business, I should say, for males. And the last thing about cats, nowadays most of the cats are fixed, right? But cats not fixed. When female goes into estrus, have you seen cat? like that is ready to get
reproductive.
Yes. Have you ever seen the cat?
Of course, I have two of them. And they're
Yes.
both
What they
murdered and
It's
safe.
my cat right now.
Yes. What they do? They
I have a cat.
Bites the neck.
Yeah, but females, what they do? They are first of all they screaming, right?
Yes.
Yeah.
They scream and they also leave their butt up, right? Yes.
Yeah.
And they are kind of like don't respond to anything. You can just grab this cat, turn upside down, and they won't do anything about that, right?
They won't.
Yes. So in cats, it's quite interesting because the cat will gonna keep screaming until she's gonna get pregnant. Now, if you ever seen male tomcat penis, it has like bunch of like tiny spikes, right?
Yeah.
So once it actually mates the cat, right? By the end of mating, he's gonna pull it out from the cat, and all the spikes kind of like open up, and they're gonna scratch cat's vagina really, really hard. It's very painful for the female cat. But what's interesting, this pain triggers release of her egg cell. So her egg cell is not going to be released until she mates a cat. Okay? So this pain will make her release her egg cell, ovulate, and here go, it's going to be fertilized by male sperm. But it's so much, it hurts so much that males, they hold their cat's neck, right, with their teeth, and they run away. Again, really fast. She's not gonna eat them like spider, but she's gonna attack them. Okay, she's gonna attack them because hurts. Okay. So something different actually from many other animals where the egg cell already released, right, at the time of mating. Okay, so yep, sexual reproduction definitely has its advantages, but there are also some tricks about that, right? Okay. Any questions here? Now, we talked about this last week, right? Human sex chromosomes, X and Y. Males, XY, females, XX. Remember, I told you that Y chromosome is much smaller than female, right? And I also explained you determination, right? Remember, I told you female can give one of her X's, males can give either X or Y, right? And I told you that it's determined by male, even though doesn't have control, right? And in other species, it's different actually. Birds have different system. I think they have X, Y, and Z chromosomes that their combination determines the gender. In many reptiles, you can determine the gender at the time of fertilization. For example, in like temperature can actually define whether it's going to be male or female. Like when female crocodile lays eggs, actually eggs that close to the surface where temperature is lower will be males, and eggs in the center where temperature is slightly higher, that's going to be females. Okay. And I also told you about hermaphrodites, right? So they can produce both male and female gametes. In turtles and crocodiles, I told you that's what temperature environment determines their gender. Okay? And guys, the last part, the very, very last part is going to be about chromosomal aberrations. Any questions so far about meiosis, mitosis, anything?
No.
No? Did I slow down?
Nope.
No? I'm still fast? Yes? Am I still
Very,
Oh yeah.
too fast?
very.
Sorry. Ah, that's like, yeah. Please remind me. Please remind me from time to time. I'll probably like I'm kind of like taking away. Okay, so chromosomal chromosomal aberration. So female, right? 44XY. So 46 chromosomes total. Because mother gives 23 and father gives 23. But sometimes what happens during meiosis, there was a mistake, right? So during meiosis, when you had 46 that had to divide to 23, 23, there was a mistake. Mistake happened. And it divided into 24 and 20. Okay. So imagine there's an egg cell that has 24 chromosomes, was fertilized by sperm cell that had 23. So the total number, instead of 46, 47, right? And remember, each chromosome has a pair, right? 23 pairs. But what happened here, that instead of the pair, you had extra, right? Instead of 2, you have extra chromosome. This is called trisomy. Trisomy have an extra chromosome in a pair. Unfortunately, it's not an advantage to have extra chromosome. It's actually a huge disadvantage. Why so? First of all, most of the reasons, like majority of reasons for miscarriages that happen within the first 3 months are actually chromosomal aberrations. Unfortunately, people prone to make these mistakes, okay? Unfortunately. And quite often, the wrong number of chromosomes in developing embryo will actually stop the development automatically and cause miscarriage, okay? At very early stage, maybe a little bit later stage. And the chance to have a child with trisomy exists, but for them, chance to survive into, into birth is not that high. Again, most of them actually don't develop into full term. The most common trisomy, the most common chromosomal aberration is, what do you see here? Trisomy 21, Down syndrome. Have you heard? Guys, talk to me. Have you heard about Down syndrome?
Yes, I have heard about Down syndrome.
Yeah.
It's an inherited condition, and it's
not inherited.
Sorry, not inherited. It's a genetic condition.
So you see, that's what I was talking about. Chromosome number 21. It's a tiny chromosome, and because it's tiny, they actually can survive into full term, right? So you see 1, 2, 3, trisomy for 21. Okay.
I just have a question.
Yes.
You can tell if a baby has Down syndrome before it's born, right?
Mm-hmm.
And they don't. Some have moderate or mild intellectual disability. They're not. They're not. It's only a stereotype that
They have limitations. They do. They do have limitations, unfortunately. Okay, but what you can see here that maternal age increases chance of having child with Down syndrome, because remember I told you when female get older, fertility goes down, plus quality of the egg cell also goes down. So meiosis is more prone to mistakes, right? And that's what pretty much is the reason of something that is called non-disjunction. Okay, non-disjunction, that's exactly what I was showing you before, right? So homologous chromosomes, right? And they didn't separate evenly. Yes. So here, extra chromosome, and here, missing a chromosome. Okay. So you see these two homologous chromosomes, instead of going opposite ways, right? They went together, right? And here you got extra, and here you got missing. Clear, guys? What is non-disjunction? Guys?
Unequal distribution of chromosomes during
Yes, it's uneven, like, and again, this is trisomy for 21, and again, having extra chromosome actually hinders a lot of cellular processes. So it manifests not only in intellectual development, but they have a lot of health issues. Usually they have very low birth weight. They have serious reflux issues. They have, quite often they have heart malformation. So, but with the good care, with a lot of love, they thrive. They thrive. They can do well at school. They can, the only major problem with that, with trisomy 21, is that they don't mature. Okay, it doesn't really affect learning. They probably learn slower than children without Down syndrome, right? But it's the thing that they can't really take full responsibility for themselves when they grow up. Okay, so that's, I think, the biggest limitation with the Down syndrome. So somebody has to take care of them. Somebody has to supervise. Somebody has to make sure that they're safe. Okay. But otherwise, I think they're very sweet. They're very nice. They're very friendly. They are very open, and okay. So trisomies that happen to autosomes, regular chromosomes, quite rare, except for Down syndrome. Why? Because larger chromosomes, trisomy would definitely affect development and again cause early miscarriage. Okay. Known is for 13, 15, 18, 21, 22. 13, 15, 18, if they actually develop to full term, right, they don't survive for more than a few months. They die shortly after birth because it's a really, really severe condition. 22, I don't know, it's more rare than 21. It wasn't described well in the literature. But it's a different story with, this is the last two slides, different story with sex chromosomes. Okay. So it doesn't affect health and like ability to learn much. Okay, so let me just go over a few of them. There are, I think, only 4 described. Are you with me, guys? Are you with me?
Yes.
Okay.
Yeah.
So
Yeah.
one more time. We just talked about trisomy for 21. Now, the same non-disjunction, what might happen? Mother will give X chromosome, right? Sex X chromosome, and father gives nothing, right? Nothing, no X, no Y, right? So what's gonna happen? She's gonna have 45 and only 1 X, okay? Now, this is called monosomy. Trisomy, extra chromosome. Monosomy is missing chromosome. Only monosomy known is monosomy for X, and that's called Turner syndrome. Now, interestingly, if there's only 1 X, right? 1 X, that's going to be female, right? So Y will make embryo masculine, right? It will make a male. But it's not actually, how to say, in this case, it doesn't matter what is missing. If she's missing X or Y, okay? If she has only 1 X, that's going to be female. The template for development is always female template, okay? So what happens if Y is present, then it's going to develop into boy. But if Y is absent, doesn't matter how many X's, it's going to be female. So Turner syndrome. So Turner syndrome is actually something like that. Okay. Sorry. Most of them diagnosed, they most diagnosed after birth. Not that many actually diagnosed prenatally. They look like girls that never reach puberty. Okay, so like really, really at the verge of puberty, but nowadays they all get hormonal therapy and they look completely feminine. They look much better. The only problem is that they can't have their own children. So fertility should be 2 X chromosomes. Without second one, fertility is greatly compromised. You can tell, I mean, professionals can tell. They usually short. They might have a lot of actually birthmarks, like a lot of freckles. And very common, they have like a fold on the neck. That's called like a webbed neck. Okay. And like the forms, as I said, like girls at the verge of puberty, okay, but not yet, not fully there. Okay. Now, sex chromosomes carry genes that actually contribute to growth spurt. Okay, so getting taller actually depends on presence of both sex chromosomes. So having only 1 X, they actually are like below average in terms of height. The next one is Klinefelter syndrome. So Klinefelter syndrome is XXY. So which is kind of like hard to say which one is missing, which one is extra, okay? So Klinefelter syndrome is something like this. Hey, what is that? Okay, here. First of all, extra sex chromosome, they are taller than average. Okay, so they taller. And again, extra X interferes a lot. So you see a bit like feminine way they built, right? They could have some breast development, and they have tendency to lose chest hair and mildly learning disabilities, which actually, interestingly, could be reversed with their testosterone therapy. Okay, so they respond well. The only problem, again, fertility. So they won't be able to have children on their own. Now, Jacob syndrome. So Jacob syndrome is XYY. So second extra Y chromosome. Okay. So second Y chromosome makes them tall. They usually prone to have acne, right? No issues really with learning and no apparent learning disabilities, but it's believed that actually have an extra Y chromosome, which corresponds to extra testosterone, can make them more aggressive. And was a lot of noise about that. Okay, so that they kind of more prone to violent, aggressive, criminal misbehavior. There's no scientific proof actually for that, for XYY. Okay, and the last one I can't really find it. Females that are XXX female, that might compromise their fertility. They usually tall. They usually also kind of maybe might be slightly less intelligent, right? But they usually much taller than average female. Okay. So what I want to say that given this information, I believe we all should take a moment